I appreciate the chance to come and talk to you about fungi. I don't want this simply to be a series of lectures where I'm talking to you. I want it to be more of a question and answer. So like in any situation, if you have questions as I'm talking, put up your hand, just, and I'll stop and talk. But what I want to do today, if you've had a chance to look at the handout, is to talk about some of the concepts that are important to you as producers or consultants or whatever your role is in the agroecology of this region, about what fungi really are. Now, I have to admit, this is going to be a deep dive into what fungi do, because you can't understand why they're managed the way they are unless you understand a little bit about their biology and why they do the things they do, or why can't they do the things that we hope they do, unless you understand really about what fungi do, how they grow, how they reproduce. And like all of us in the room, not all of us have the same capabilities, right? Not all of us have the ability to do certain things. And fungi are the same way. And so what I'm going to hope to show too is that while you may have fungi in your systems, there are things you're doing or not doing that are keeping the ones that can do more out. And that's based on the biology of the system. You would not want a room full of John Zacks. Nothing would get done, right? Because you need a diversity of people and approaches, and the same way is true for fungi. So, like I said, I've scheduled it today. We have Q&A scheduled after, but anytime as I'm talking, if it's not clear, put up your hand, I'll stop and we'll talk. But I'm going to look at what fungi do, how they're structured, how they grow. That's the most important thing. How do they really grow in your soils, in your fields, on those roots, and then what happens as we manage that system. And then I'll talk about the important components of the mycorrhizal relationships to plants, what fungi are involved, but in more detail about how that system, as we know it now, actually works. Why does it work the way it does? Are all mycorrhizae created equal? And the answer is no. So we're going to look at that. And then that's the morning. And then the afternoon, we'll talk about this whole notion of carbon and litter and decomposition and carbon sequestration. We'll get into looking at this whole notion about how do you build soil carbon and what are important. Are bacteria important or are fungi important? What's the relationship there? And then part of that is understanding what you do as we manage soils. What happens when you apply pesticides or herbicides? What does someone ask me this morning? Well, what happens when you apply fertilizers? Is it all bad? And like anything else in life, yes and no. Right? It depends upon how much you're doing and what you're going to do. And I want to talk to you then about how fungi respond to the fertilizer that gets added, and why do they respond the way they do. And then lastly, I'll try to wrap it up by talking about, okay, having known all that, where are we with fungi in this whole region? What do we know about what's going on in these systems? Again, if you've been to the Symposium and talked to them, and you listen to the people that have talked, Dr. Lassell, Dr. Zimmer, over the last couple days at the Soil Health, their systems and where they've done are not the same as ours, and the fungi here are going to respond in much different ways than what happens in Wisconsin. It has to, because they're a different group, right? Different, and so we're going to try to understand that. So at any time, if it's not clear, just raise your hand. Say, and by the way, it is John Zack. I wasn't born with doctor, so right. So it's John. Thank you. And as I found out today, I teach non-majors microbiology and some majors. That's my fall class. And then I do upper division classes in fungal biology and microbial ecology. And what I've discovered today is two of you sitting in this room beside RN, I've had in class. And I said, oh my God, I'm getting old, right?
It's because we look old.
Okay, so let's start this process. Now, one of the things I think is important for us to understand is that probably within the last 20 to 25 years, there have been more and more people being aware of what fungi do on the planet. why they're important, what do they do. And I picked two articles. This is Michael Phillips in Mycorrhizal Planet, how fungi actually benefit. And it doesn't matter whether you're growing forests or whether you're growing grasslands or whether you're growing cotton. What do they do? And then I like if Kyle Kazaniski. Have anybody seen his YouTube videos? Oh, he's great, because what his whole discussion is, how do you farm with fungi? How do you actually farm with fungi? Now again, he's not in West Texas, as you can see by what's being grown. He's someplace else. Now, let me ask you another question, because it helps me. How many of you have had any plant pathology class? Okay, so unfortunately, I'm not going to talk about any plant pathology, right? Good, I'm talking about the other stuff. How many of you have ever had a mycology class? Anybody? No mycology? That's the study of fungi. Okay, so I know where I'm beginning. Okay, but I point this out that in the last 25 years, this has really taken off. How many know who Paul Stamets is? Anybody know who Paul Stamets is? Paul Stamets is a mycologist up in Oregon that has put out a lot of information about the role of fungi in natural systems and how we as humans are not using the products of fungi to actually do more. And so he's pushing that pretty much, and has been for about the last 20 years. But I want to show you something. We're not the first group to ever do this. The role of fungi in human society goes back centuries. Right? And it was the Greeks that actually started working on fungi when that civilization developed, right? And people knew because mushrooms were so common and people saw them, what was going on, right? I think I've mentioned this before. How do you know what mushroom is good to eat? Somebody had to die. Somebody had to die. Somebody said, I think I'm going to try this, right? And they try it. And what happens? They get sick, okay? That's the worst. And some people eat them and die within 24 hours. By the way, I should tell you, if you don't ever want to go out and just collect mushrooms and eat them. There's no cure for high mushroom poisoning. You end up destroying your liver and you're dead in 36 to 48 hours, right? But we had to learn that, right? We had to learn that. And then this whole notion about fairy rings. Why do you see rings of mushrooms in your yard? And and some mushrooms were edible and otherwise they were toadstools. Look at the date. 1526 was when that book was published. The fungal pharmacy. And then of course, humans being what humans are, everybody likes the hallucinogenic stuff, right? So what roles do the hallucinogenic mushrooms have in our societies? Psilocybins, Amanita. At lunch, I'll tell you a story about an Amanita issue that occurred here in Lubbock. Now, we also know that there's a lot of products that we consume that are really only made by fungi. And we've been doing this for centuries, right? We know they're important for organic matter decomposition on the planet. We're starting, and basically this information did not start in that top point until the middle of the 1930s. That's when we started to connect up what fungi are really doing, right? But we also know about plant pathogens, right? We've known that for years. We didn't know why. And then the whole issue of animal and human pathogens, right? Scabies in sheep, athlete's foot, coccidioides, all these things that are going on around the world right now. I don't know whether you know this, but the world is not just in a COVID-19 pandemic. The world is actually experiencing an enormous rise in fungal human diseases as a consequence of our movement around the planet also. And we're bringing fungal diseases back in ways we've never thought possible. Now, I have to tell you, you don't ever want to catch a fungal disease. And if it's on your skin, it's one thing. If it's in you, you're in trouble. And we'll see why. And then look at the products, soy sauce, citric acid production, beer, wine, mushrooms, and bread. They're all fungal derived. And we had to learn that over time. And then antibiotic production, penicillin, cephalosporins, Grisiofulvum. And then the one thing that's also important for today's lecture is this third component. Our microbiome that keeps us healthy is all bacterial. We have no fungi in us, and we don't want fungi in us. Our gut microbiomes are all bacterial derived. But plants don't have that. Plants have a microbiome that's dominated by fungi. And those fungi are in the leaves, they're in the seeds, they're in the shoots, they're in the roots. And if you don't allow that to happen, what do you end up doing? You have to fertilize up the wazoo to make up for the fungi that you did not allow to form with that microbiome for that plant. And that's where this continuum of how fungi interact in our world, the two major ones, decomposition of organic matter and the microbiomes of plants, plays the major role in any ecological system, including agricultural systems. The question is, in agricultural systems, do we allow it to happen or not? Now, this is where it's going to go deep. So really, what are fungi, right? Where are they in the whole schema of life? Well, it turns out that in actual fact, there are two groups of fungi. There are fungi that are in the kingdom fungi. This is the kingdom for bacteria, plants, animals, protozoans, fungi, and then there's this other group called Chromista or Stramanophilia. Fungi are in two different groups because they've evolved at least twice in different ways. So you have, everybody knows about Fusarium, that's why I picked it. So we have Fusarium in the kingdom fungi, and then you have Pythium in the kingdom Stramatophilia. Root rot, Pythium root rot, damping off of seedlings. Those are fungi, but they're not the same fungi as the Fusarium. They're not the same fungi as the mushrooms you go to the store and buy and eat. They're completely different. But they respond in similar ways. They look similar. So that's why we study them. Okay, so what we're going to look at is sort of their growth, ecology, and structure, and try to understand what fungi do. And let me get my phone here so I can see the time. Okay. Okay, so this is where it's going to get complicated. But you have to know what fungi are. So I have a picture of a puffball here. Is this going to work? Yeah. This is a puffball that's puffing its spores, right? And that's what happens at any time you have these things out in the environment, right? Now, the majority of fungi are filamentous. That means they are strong, they are long strands of something that grow as a filament. I was in a project one time where we were looking at the length of filaments that occur in a gram of soil out of a forest. And so a gram, you take a spoonful of sugar, and that's about a gram, maybe a little more. So you have a spoonful of sugar, and that's how much soil you have. And the question is, what is the length of fungi in that 1 gram? Anybody know? What would you give me a guess? How many? Miles? How many miles? Yeah, how many miles? Well, it turns out that in a normal productive forest, in that top couple inches of soil, there are 4.5 miles of fungi in terms of length. So if you took all those filaments and put them end to end, you would have 4.5 miles. So I want you to go home and get a roll of the finest thread that you can do and measure 4.5 miles and see what that volume looks like. And that's what you have in the forest soil. Now, how much do you have in a good agricultural field? You have anywhere from a 1/4 mile to maybe 6 miles of fungal filaments growing in that system. And the challenge then, yes, sir?
How much do we have in just plowed fields forever? Not good farmland, what you call it.
I've measured some. The worst I've seen around here is less than 10 feet.
Thank you.
Yeah, less than 10 feet. Yeah, there's not much. And there are methods to be able to extract that and measure that, right? Yeah, I mean, and that's part of the problem. Because we don't see them, you don't ever know how much you have, right? But there are ways to do it. Okay, now, they're filamentous except for the yeasts. Yeasts are fungi too. They're not filamentous, they're single-celled. Now, the other part is that they're eukaryotic, which means they operate just like us. They're not any different than us. The fungi are our same cells that make up us, that make up plants. It's the same. They're all heterotrophic, which also means, like us, it would be nice, and I've always wished this with God. I wish one day I could just walk outside, spread my arms, photosynthesize, and get all the energy and carbon I need. That would be terrific. I wouldn't have to consume food. Now, I'd miss it, but I wouldn't have to consume it. Well, fungi have to eat also. They don't make any of their own food. They have to get food that's already been made by plants or by something else. That's what it means by heterotrophic. Now, they're multinucleated. They are colonial organisms. They grow like corals. If you don't realize that, corals are colonial. Fungi grow like corals, except they're filamentous. They're indeterminate, and that's important. Fungi can grow forever. They can keep growing. There's no limit to how big they can get, except they're damaged or they're killed. They're also modular, which means that we're not modular. I can't chop myself up and regenerate me. I can cut up a fungus to some degree, and it'll get spread around, and it'll reproduce. That's why you can take cuttings from plants. Plants are modular organisms. I can take a cutting and root it. I can do tissue culture on a meristem of a cotton and regenerate cotton because plants are modular. Fungi are modular. They get spread around that way. And then lastly, they're haploid for most of their life cycle. We are diploid organisms. We have two chromatids for every chromosome. Fungi initially only have one. And so they have a very different approach to what their life is. Now, the reason that this is important is that this whole nature of filamentous, eukaryotic, and heterotrophic plays into the whole notion of why they decompose, because they have to get their carbon. But these modular and indeterminate also comes into play as to how they interact with the environment, but also how they interact with the plants that are in that environment. Now, this is force of habit. Any questions? Okay. Now, what do we know in general about fungi? Well, right now, there's about 80,000 described species of fungi around the world. There's probably 8 to 10 times. They're found in any terrestrial environment, all freshwater environments. Notice one thing, there's no marine fungi. They're not found in marine environments. They reproduce by spores, either asexual spores, which are clonal, or sexual spores, decomposers of nutrient matter, nutrient cycling, and then some are plant pathogens. So here we have mushrooms decomposing the wood. We have this rust on apple. That's the fungal structures you see here. And then these rings. Anybody know what these rings are? That's ringworm. So that's a fungus growing in your skin. That's what it is. And that's what they look like. So by the way, if you have athlete's foot, it's a fungus growing between your toes. Right? And it's the same thing. Okay. So that's what I'm talking about in this whole notion of being heterotrophic, being absorptive, diffuse branched tubular body. And that describes how fungi live in the environment and what they're able to do in terms of providing the capacity for that ecosystem, whether it's agriculture, a forest, or natural system, to actually carry out the functions that fungi do that have evolved. Now, the one thing you need to also keep in mind is that fungi are prolific spore producers, right? And these are examples of spores. Let me see if this will work here. There we go. So these are the types of spores that fungi produce, and their primary method of dispersal is two. They either blow around in the wind or they move in water. And so all fungi begin by germinating as a seed, like a seed, from that spore. And so each fungi has what's called a germ pore that germinates from. Could be that in there, could be that. And then it produces a single filament called a hypha. And then that hypha collectively is called hyphae. And then when you get a mass of those filaments, we say it's the mycelium. So what's growing in your ringworm on your athlete's foot, what's growing right now in the block of cheddar cheese in your refrigerator that's going to get moldy is the mycelium of the fungi. You don't see it normally. It's too small. What you see on the cheese are the spores that it's producing to disperse itself. And all fungi at some point in their life cycle are going to produce spores. Now, as I'm talking in this last minute, every single person in this room took a breath. How many spores of fungi did you breathe in? In the average human environment, it's 22. You just breathed in 22 spores of some fungi. And the question is, why aren't you killed by fungi? Right? So think about how many breaths you take over a day, particularly when the dust is blowing in Lubbock, you're picking up a lot of fungal spores. But you don't die of that, right? Because our immune system, our respiratory tract keeps that going. Now, I will tell you one thing. Not now, but previously, the individuals that came down with AIDS, that virus that affected their immune system, what they died of primarily were fungal infections. Almost 98% of them. And the reason is the virus damaged the immune system to such a degree that they could not keep themselves from being colonized by fungi. And that's how the AIDS victims died. And so, we talk about the wondrous nature of what we are. It's amazing that none of us are killed by fungi because we interact with them all the time, right? We interact with them all the time. But that's also why I've often said that, if you're if you don't necessarily have to add things to your fields to get things to happen, because these fungi are producing spores everywhere that blow around. They will find what they want. They will find what they want, and they will colonize that material. But it's important to understand that fungi start out as this mycelial structure. Right? And I put some pictures of spores. Those of you familiar, this is aspergillus. So we talk about corn mold. That's one of the fungi that get on corn and produce aflatoxins. So we have aspergillus, we have a mushroom produces spores, and then this is what a yeast looks like if you put it under a microscope. It's this bowling ball structure, right? That's what made your bread this morning. That's what makes your beer or your wine. It's those organisms interacting. Now, you have to also keep in mind that fungi, unlike us, are unique in a couple ways. A lot of fungi have what's called an asexual stage. That means they grow as mycelium, but they keep reproducing themselves clonally. It's as if we're sitting in this room and every one of us over the next hour produce 6 copies of themselves. And they're identical copies of themselves. And they go off and do other things. So a lot of fungi reproduce that way in this sexual stage. It's called the anamorph. But a lot of fungi also have what's called a sexual state in which, like us, they have recombination. So we produce eggs and sperm, and we produce our next progeny. Fungi will do the same thing. They'll exchange nuclei, produce a sexual stage that then produces other spores that go and regenerate their progeny. And that's what mushrooms are. Mushrooms are the sexual stage of that fungus. That's what we eat. We eat the sexual stage of the fungus as it produces spores. So this is what fungi look like as they grow. How many people have seen pictures like this? Yeah, a few of you have. Okay, so again, we start out as a spore, it germinates, and you'll notice that it keeps branching, and it grows in a circle. And so we have this mycelium that is growing through the environment, your soil, your litter. It's growing on your plant, it's growing on the stubble that's on. But what is it doing? It is searching for resources. We move toward our resources. Fungi do too. They simply grow toward that carbon, those nutrients, until they find it, and then they start taking it in, and they absorb it. Fungi have no mouth parts, as we see. They have to produce enzymes. They excrete that then break that stuff down. But, by the way, what is the shape of this? It's circular, right? That's why the ringworm is circular, because it started out as a spore that simply grew out. So anytime a spore lands in your field, on your plants, on your litter, in your soil, it's growing out like that. It's moving in that circular pattern. And this is an example. This is a fairy ring. This shows you the circular pattern. These mushrooms are fruiting at the edge of that circle. That's how they grow. But that's what's going on anywhere. And the question then is, so as you plow, or as you go in and do strip till or minimal till or whatever, you break all this up, because this stuff is not very deep. It's in that top couple centimeters growing. Here's the largest fungus we know of on the planet today. This is Armillaria amelia. It's a mushroom. It's been estimated at, 8,700 years old. That's how old it is. It covers, that mycelium covers 4 square miles, 2,384 acres. And if you were to weigh it, it weighs about 650 tons. But what weighs? It's the mycelium underground that you cannot see. So if you said to me, John, do I have mushrooms in my fields? Yes, you do. Do I ever find them? Rarely. Why? Because they don't produce the teleomorph. They don't have to produce the mushroom all the time. They do that when they need to. Now, I have to tell you that RN has some really good mushrooms in his field. They're not hallucinogenic, but there's some really good mushrooms. Right?
Would they kill me?
No, yeah, I wouldn't eat them, right? I wouldn't eat them. They may make you really sick. You may wish you were dead, right? But they wouldn't kill you. But every single person that has a field, that has a yard, that has a garden, you have mushrooms in it, whether you know that or not, in some way, shape, or form, because these organisms are part of that dynamic of dealing with that carbon in that environment. But again, you don't know that because most of the biomass of what that fungus isn't this. It's the mycelia that's below ground in the litter, and that's part of the challenge for understanding how to manage. Now, how do these things really grow? Well, as I said, they are filaments. And so what fungi have cell walls, just like plants do. And they have nuclei and everything else that's important for their cells. But where they grow is they grow at the tip. So it's like a big balloon that you keep blowing into. You know, as a kid, I liked those balloons that were ribbed that you could blow up to 6 feet long, right? And you just kept blowing into them, and they keep getting longer and longer and longer and longer and longer. But that's important because the fungi grow by extending that tip and growing forward. And then they also have these things called septa, which help them structurally keep rigid, but they also have pores that, in case something breaks. I mean, it's like a straw. If you break a straw, everything's going to flow out. If you break one of these, things flow out. So they have mechanisms to keep themselves from being damaged. But again, what damage do you primarily do? You till. I dig. When you walk across your yard and it's wet, right, you break up this mycelial network by stepping on it. So a nice drawing of what these things really look like inside, right? So I'm sure we've all seen pictures of what the cells look like to us, but here we have the cell walls. Here we have all the cellular parts that keep fungi growing, and then you have the structure at the tip. I like the term. The term, it's labeled as SPK, right? It's a German term. It's the only German I ever speak. It's called a Spitzenkörper. And what it is, it's a collection of vesicles that allow the organism to produce this tip. Now, I have to tell you something that some of you probably don't know. How does cotton fiber grow? You all grow it. How does it actually grow off the seed?
It's a single cell, an extension of cell wall, and it's one single cell.
That's right, and it extends. So guess what cotton has? It has a Spitzenkörper. It has a collection of vesicles at the tip that determines how long that fiber gets and why that fiber looks the way it does. Something has to organize it. It's just not haphazard, right? You just can't put all the stuff in a cell and expect it to organize itself. So fungi, like fibers in a cotton, off a cotton seed, have this structure at the tip that allows that cotton fiber to organize the wall, that cellulose wall, as it grows out, and to keep it integral to the length of what it's supposed to be. And fungi do the same thing. They have to control what they grow. How does a fungus know where to grow? It does. How does a cotton fiber know how long to get, where to be? It has to have an organizational structure at the cellular level that does that. Now, I've mentioned that fungi are heterotrophic. They have to get carbon. And these are the 4 ways fungi get carbon. And what's interesting is that fungi can be some of these. There's no one fungus that can be all of them. The majority of fungi are decomposers, saprophytes, and that's the largest group. Those are the ones that live off any dead organic material. The plants, the hair we lose, the skin we slough, the animals that die, us when we eventually die, all that carbon that's in us, and that gets decomposed, and they use it to build their own biomass. But that's what keeps the planet functioning. And then we have the fungi that we call lichenized, and so they're associated with cyanobacteria, green algae, to form lichens. Lichens are a symbiotic organization, as we'll see. And then for us, the mycorrhizal fungi, symbionts with plant roots as part of their plant microbiome. In the same way that if I removed your microbiome, you'd be alive, but you wouldn't function well. If I reduce or remove the mycorrhizae from plants, they don't function well either, the ones that do form it. And then lastly, the smallest group are the pathogens. Interestingly enough, like bacteria, the number that people have given is that less than 0.1% of those 80,000 species are pathogenic. The rest fall into these 3 categories as to where they get their carbon. Now, some fungi are decomposers and lichenized. The mycorrhizal fungi don't decompose. They're strictly part of that integral microbiome. So how do fungi grow? And I mentioned they grow at the tip. So what happens is the fungi grow along and they will produce a branch. So what happens is they have to generate a new Spitzenkörper to be able to branch. And so they will go along and they will produce a new set of Spitzenkörpers until they form this network of mycelium. And they do that for what purpose? They do that to increase the surface area over which they absorb stuff. That's how they grow. Again, you know that already because that's what roots do. Your cotton produces laterals and primary laterals, secondary laterals, taproots, and what are they doing? They're branching to be able to absorb more out of the system. Well, fungi are able to do that in a much more extensive way. They will simply keep growing and growing and growing and branching and growing toward that substrate. Now, here's where the rubber hits the road in terms of tillage. Every fungus has what's called a hyphal growth unit, which is the total length of the mycelium over the number of hyphal tips it has. If you break up a fungus in any way, shape, or form, right? It has to have a certain hyphal growth unit to be able to recover. So the more you till, the more you dig, and you break up this mycelium, depending upon the fungus, right? Depending upon the fungus, it doesn't recover because they all have certain size hyphal growth units. So I've put a number here. In fungi, it's between 30 to 400 micrometers. So in other words, you need a certain number of tips and you need a certain length to last that damage in order to regrow. And you'll notice one thing, and this may be why we don't see a lot of mushrooms in fields. Basidiomycetes, which are your mushrooms, have the highest hyphal growth unit. They are the ones that are most susceptible to damage. They simply don't recover. Now, this is also one of the reasons why in commercialization of mycorrhizal inoculum, there are certain things that work and certain things that don't, where you have basidiomycetes, because these fungi simply don't like to be broken up. They will not recover. Now, what about how do they capture resources, right? How do fungi actually grow? So we see this enormous mycelial mat. One of the things that happens too is as fungi grow, think about this, and they branch, they produce new tips, and they keep growing. As they grow out to a resource that they want, what they'll do is they'll branch even more, because what they're trying to do is, like roots, they're trying to increase surface area over which they can absorb and use that carbon and nitrogen and phosphorus and bring it in. So as fungi grow, they may not produce much mycelium until they get to what they want, and then they'll simply explode and produce all this network of mycelium because it's increasing surface area. And, I put this up here. This is bread mold. These are bread, and these fungi produce these rhizoids, but it's like tillering in plants. They simply produce a main group of spores. They'll jump and produce a rhizoid, or a stolon, produce another one. So fungi have different mechanisms and growth rates in the same way that grass as well. I like the picture yesterday where it was the sorghum, I guess it was on 80-inch centers, where some of the sorghum planted on 40, it never tillered. Those that are on 80-inch tillered. And they produce all these heads. Fungi do the same thing because it's a common ecological response to what? Nutrient capture and environmental resources. They have to be able to do that. By the way, this picture here, where you have a very tight group of structures. Anybody know what the military term for that form of movement is? And this is interesting because it's in the ecological. This is what's called a phalanx, right? So when you want to really go into something, you put a lot of people together, short knit, short tight group, and you simply move through. And that some fungi have that, right? They have that growth form where it's so tightly knit, nothing else gets in. And they'll colonize that. Where this is what's called a guerrilla form of colonization, where you're sort of jumping around all over the place. Again, I put this up here to show you how the extensive network of fungi, as they produce these cords and wafts of mycelium until they get to some place they want to start using resources, and they finally branch. By the way, this is what our nervous system does, the same thing when it innervates muscles. You have a single, right, we all have a sciatic nerve that runs out of our back down our leg until it gets to some of our muscles, and then it expands and multiplies and innervates that muscle to cause our leg to move, right? And so fungi are constantly rebuilding and moving. Nothing in the fungal world is static. So in the entire growing season, these fungi are growing and moving, and they're never in the same place in your field. They are constantly moving around looking for carbon and working on that system. Nope. Let me go back. One of the things I think is also interesting is that, in these structures here, in these cords, you actually have fungi building what look like conduits of large tubes that are highly organized. They almost look like multiple blood vessels. But these are fungi. And do you know why? Fungi translocate water. They move water from one source to another. They will move water. Now, not all fungi do that, but the basidiomycetes, some of the ascomycetes, produce these large structures. And I've seen them in some of the fields that I've been around in cotton, where you find these mycelial cords, and these are basidiomycetes primarily, that are moving water around in your system. They're moving water to themselves, but they're also moving water to the material they're trying to decompose. And in that water is what? It's all your nitrogen and phosphorus. They're moving that around. Now, so what are the impacts of tillage, right? So the problem is, it's this destruction of the mycelial network, and it reduces fungal growth and the capacity of your fields to build and store soil carbon. The problem is this: when you damage that mycelium in any way, they repair it. But they repair it by using the carbon that's in your field, because that's what they're doing. They're using the carbon in your field to repair the damage, right? Now, here's the other part, and we'll show more about this later. The fungi that are in your field that are there, and in terms of how they respond to tillage, right, are there because they have hyphal growth units. They can be chopped up. But what we discover is they're not very good at building organic matter. They may be in your field, and they may be growing, but they're not the ones you want. Yes, sir.
So
Steve.
even in a no-till situation, with the colonies and everything, just from my perspective, it reinforces the fact that we need to see continuous traffic, always driving the same path in the field, because you reckon just the tractor driving across the soil will damage.
Yeah, because what it does is it compacts. And these things are susceptible to breakage.
Okay.
Yeah. And I think I think it was you talked about the other day about just staying in one, always on the same track, same part of the field. Yeah, I mean, I've learned over the years, I don't dig my garden anymore. I try to be as less invasive in terms of just changing anything in that soil system that I can to keep the mycelial network intact, right? Now again, I'm only working on, a 50 by 50 garden. You're working on 5,000 acres, right? So, I mean, but the process is all the same. You want to maintain that network as much as possible. Now, obviously, when buffalo ran through this area, they broke that up also, right? And so natural systems, so the fungi are designed to be broken up. The problem is you can break them up once or twice, and they're going to recover. It's when you keep doing it year after year, after growing season, after growing season, after growing season, do they start to fall apart and never recover, right? I mean, yeah. I mean, as we all know, those of you that play basketball or any sport, when you're younger, you can play much more than when you can when you're 70, right? Because your body can't take the abuse, right? It's too much hitting, too much this, too much that. Well, same thing's true for the fungi. You can do it periodically. You just can't do it a lot. And again, I think I was talking yesterday, and there are no numbers on this. In a given hectare of field, what's the minimum disturbance that you can do and not change things much in that level? And the problem is we don't really know, right? But the advice is do the minimum on that. Yeah, good question.
Okay,
Martin.
So you're talking about building organic matter with the fungi is obviously very important in that process. But you take areas like, say, in Indiana or whatever, if you looked at their organic matters, they can get 3 to 4%, whereas here, even under continuous no-till, and when we're doing things we think are what we need to be doing and correct, our organic matters aren't getting more than 1.7.
Right, or 2% at most. Right.
Right. So what is that a soil structure thing for our area, is that fungal related?
No, well, no, it's a combination of everything. Basically, what I know of and what the literature and research has shown is this. The reason we don't is we have a much longer period over which the fungi are active to decompose. And so let me give you a classic example. If you go down to the tropics, their soil organic matter in a tropical rainforest is about 0.2%. But look at the size of the vegetation that gets supported and how diverse it is. And so the question is, well, why is that? Well, that's because the microbes, the fungi particularly in those systems, are always working on the leaves and stuff that get dropped and turning them over. So they reach that equilibrium. The reason we can build more soil carbon up north is it's colder, and 6 months out of the year the fungi don't do anything. So the soil carbon, as we'll see, has two components. It has the plant material that is produced, but it's also the material that the fungi work on over time. And here, because it's warmer longer, they simply have to deal with the temperature and drought issues, and so they use more of it to do their repair. Now, having said that, our systems naturally are going to run about 2%, maybe 3%, right? But that's the climate equilibrium. And the fungi that you have are always building toward that. But when you don't have it, that's when you see the 1%, 0.8%, 0.4%. No, most of these fungi will not grow much below soil temperatures of about 40. They'll start to slow down pretty good. They just don't like to be active at that. It doesn't mean that they're damaged. It just means they're not active. RN.
Their cellular structures, when they freeze in these colder regions, it doesn't kill.
It depends. No. And so what happens is, with some of these structures, fungi will produce a lot of glycols and things to keep themselves from freezing. I mean, that's how trees work, right? They'll produce these things to keep their height from being damaged by ice crystals, right? And so that's what they'll do. They'll overwinter. Now, some fungi produce spores, so they may simply, some of them that, see, you have to keep in mind that there are what are called weedy species of fungi, and there are species of fungi that are more ecologically efficient. And you have weedy fungi, and so the problem is, you're going to have the same level of fungi in one field versus another, but one's going to be a whole field of weedy fungi, and the other's going to be a whole field of beneficial fungi. But the problem is when I go to measure how much, it's going to be the same, because it's the environment that's controlling it. I will tell you this, my graduate students get so fed up with me, they can never see a difference between irrigated tilled cotton and irrigated no-tilled cotton, even with rotations in fields, in terms of the fungi. They said, look, Dr. Zach, we always have the same amount. And I said, yeah, because it's responding to the moisture and the climate constraints. But are they the same fungi? And the answer is absolutely no. And that's what's different. It's the functionality difference that makes the difference. Yes, sir?
Think a guy plows every year, and then another guy plows every year, and puts 30 tons of manure out. Is that guy that put the manure out going to have more fungi, or will that heal that ground up more than the guy that didn't put the manure on? Does the manure have any impact?
The manure will slow down the deterioration, but it will not stop it.
Will it help the fungi?
No. Only for that year. Because then it's like Groundhog Day. You keep going back to time zero. Every time you plow these things, you go back to time zero. Doesn't matter what you do in the interim, you go back to time zero.
Should you plow manure in or just let it be on top?
I'd set it on top. I wouldn't plow it in. If you're gonna, now again, you have to sort of take this with, you have to sort of look at the larger picture. I mean, if it was me, and again, we have to run this experiment. I'm not saying that, there are some reasons why you may do some plowing. But if you plow every year or a couple times a year, that's not what you want to do, right? Now, you may put manure on, and don't plow for 2 or 3 years. You'll get a benefit, right? But again, the problem is when you go back in and plow, you reset all that stuff, no matter what. It gets reset because it's a disturbance that is major. Yes, sir, in the back.
So we talked about the freezing aspect, and yesterday somebody mentioned they measured the soil temperature at 130 degrees. So at what point does it affect the fungus?
No, so what happens, that's a good question, because I've done a lot of work at Big Bend and up here, and you're right, soil temperatures will get 130 degrees Fahrenheit, which is about 60 degrees centigrade. If I take any fungus out of your field and I grow it at 60 degrees centigrade in the lab, it will not grow. And in fact, if I give it 40 degrees centigrade, which is about 96, and I do it continuous, it will not grow. However, if I grow a fungus at the daily temperature range that soil sees, from midnight to midnight, if I grow that fungus and start out at a soil temperature of 75, it'll start to grow, and then what happens is when it reaches the temperature of 130, it'll stop. Now, if it's not more than 4 hours normally, it'll recover. And so as the temperature starts to fall back down, it'll regrow. The problem is, and that's what happens in any desert, in any arid system, any semi-arid system. If you have bare soil, the temperatures are fluctuating like that, you're not going to eliminate the fungi because nothing is ever at 130 degrees for 24 hours. It's going to go back down. But the problem is it has to repair the damage from the temperature. And what's it going to do? It's going to eat up all your soil carbon. It has to do that because they're heterotrophic. And that's the problem with tillage. You're not only damaging the fungi that do survive have to use up all your organic matter, all the manure you put on if you have bare soil. Because those temperature extremes, again, it's not doing anything that any organism doesn't do in terms of fungi. Now, up north, you don't see those temperatures. That's why you can build so much more organic matter, right? You don't see 130 degrees. It's just in these arid, semi-arid areas we do. So anything we can do to reduce the temperature variability and to keep those temperatures lower, you are going to build organic matter because it doesn't need to use it. It's going to do what it has to do. It's not dealing with the stress. RN, you had a question?
Yes. So we try to concentrate on practices, not products. And there are, I'm sure there are products that can help people, but the problem that all farmers have is we're looking for the easy solution. We want the bugs in a jug. We want soil health in a jug. We all have, and I don't know who popularized this, but I love it. We all have a moron complex. If a little does a little good, then we'll just put a little more on it. It'll do better. And when John, who you know, I love you, John, you said that about the manure. I don't know why, I can't explain why. I've heard a lot of soil health people warn about overusing manure. You can really hurt your land by thinking that, well, I'll just manure my way out of this. And I'm sure Dr. Zach knows why, I don't, but it goes back to our moron complex.
Yeah, no.
Be aware of it.
Yep.
I mean, that's a reality for me though, because I've got areas that put 30 tons of manure a year out, plowed in, and the organic matters are over 5%.
Sure, right, because they're putting it all on.
It's toxic.
Yes.
You can't grow anything.
No, that's exactly right. Yeah, because again, I'll get into this afternoon, but part of the problem of putting on manure or any fertilizer is fungi are also susceptible because of the fact that they have to absorb. There's a process called osmotic control. And so when you add salts of any kind to soils, you change the osmotic concentration of the environment in which they are in. And I'll give you the best example. I never water my houseplants with Lubbock water. Lubbock water. It's too much salt in it. right? And so eventually, my plants will have enough water, the soil will be moist, and they're going to be dead, because the osmotic concentration of the water is such that the plants can pull water out. Well, the same thing happens in your field. The fungi have to absorb water by osmosis in that mycelium. And when the nutrient concentration in the soil, the salts from ammonium nitrate, any kind of fertilizer is too high, it changes the osmotic concentration, and your fields are wet, and those fungi are dead. And that's one of the direct effects of manure or fertilization. You have to balance that. And I have some data to show you that in terms of what happens. But to finish up this, I just want to show you some other things. What did I say about destruction of your fungal network? Overwatering is one of them, because fungi are all aerobic. You keep your soil saturated, they're going to be dead. That's what happens when a field floods. Over-fertilization, as we'll start to see, because of that osmotic effect. Now, there are some fungi that can deal with that and some fungi that can't. And then the use of fungicides, pesticides, and herbicides, because again, I mentioned this yesterday at the workshop, a lot of these compounds are fungal hormones. That's how they're derived. I don't know, any chemists in here? I'm sure you know where all the products are on, but a lot of hormones for us, for fungi, for plants, are based on the amino acid tryptophan. They all have that ring structure about them. And so they all mimic fungal hormones for growth and control. That's why they work on plants too. They mimic plant hormones. And that's how they kill them, right? They mimic what the fungal hormones are doing, and then, guys, if I gave you all estrogen, I'd turn you all into females. Right? Well, that's what you're doing to the fungi. You're giving them these compounds in quantities that are changing them into something else, and it kills them. And that's the problem.
So the seed treatments we're getting, they're not targeted.
Are not what? I'm sorry, John?
They're not targeted to the bad.
They're targeted to anything. Yeah. The seed treatments that they put on seeds, they're just, you can't differentiate different fungi. So I don't know so much about that. Again, is it specific? You can't make a pesticide or fungicide specific enough not to have indirect effects on something else. So let me mention something. All of us will take penicillin from time to time to kill certain types of bacteria. And fungi make it. If I boost the concentration of penicillin in a plate, I can kill fungi with it. Even though the fungus is making it, if I change the concentration of it, I can kill the fungus. I can kill the fungus that actually makes it. So that's the problem with all this stuff. All these biologicals that we make have indirect effects at different concentrations. And so we don't have enough information to know what it's doing exactly. We know in broad terms what it's doing. And that's the level of difficulty. Okay.
The temperature, if you don't mind to address for a minute. In my younger years, there was a theory of a cotton plant on every square foot, shaded ground. The stripper header became popular for a while. Now we're evolving into wide row. Your thoughts on distribution of plants?
We were talking a little about that yesterday. It's something that that's going to have to be looked at. If you have stubble on between those plants, you're fine. If you have bare soil between those plants, you're going to have some problems. Because it's around here, it's just, when I go, Gary Zimmer and at the farm we have in Wisconsin, I don't worry about that. Right? My soils don't heat up that enough. In fact, my problem is they stay too cool over the growing season, because where our farm is, I can be 92 degrees outside in July, and I will go down to 42 degrees at night because of the nature of the landscape in Wisconsin. Stephen.
Another question on fungicides and pesticides on the seed. We're learning that the first several hours of that seed being sprouted
from what I know, from what I've seen, the root grows fast enough out of that effect of that seed coat fungicide not to have any deleterious effects on beneficial fungi. It grows too quickly because the fungi are not just around where the seed is planted, they're in the volume around it. So, I mean, really, that seed protection is just to get that root radical out without being colonized by other fungi. Yeah, that doesn't seem to be a problem. It's where things are put into the system that are more widespread, you know. Sir?
Okay, just in general, like, you know, we kill things to make a living kind of deal. And it seems like everything we do, you have to hit the target to kill it. And so, fungicides, pesticides, herbicides, all are gonna hit probably more of the surface of the target, except for things that are maybe going in furrow. And your plowing cultivations are just taking out a huge target. So my question is, how deep are most of these fungi in your soil, and can you bypass killing a bunch of these by water solubility of your products, that you are tying things up higher on the soil?
No, good question. So most of the fungi that are important for decomposition, for symbiotic relationships with the plants, are in the top 8 to 10 inches because of how we grow plants and how the roots grow. And that's true, too, of a grassland. I mean, grass plants, as you know, produce long roots, and they will move the organisms with them as they grow down. But most of that's in that top, maybe 12 inches at most. And here's the reason. Fungi are all aerobic. As you go deeper, you have problems with movement of oxygen deep down. And so they tend to stay where they're well oxygenated. That's the problem. Someone else had a question? Yeah.
So you're talking about the overwatering, and I think it was Chris that was talking about he holds off trying to water in the summer with his
Right.
cotton. If you hold off and you're making those roots look out, can you also force or encourage those symbiotic relationships? Since a lot of times a plant will send out a signal and that mycorrhizae will bring that water in.
Yeah. So yeah,
A larger symbiotic relationship.
Well, so no, it is, and part of this is if you have a good mycorrhizal network, they're working back and forth. Now, part of the difficulty too in all this is that not all mycorrhizae are capable of moving a lot of water to the plant, right? Some of them do it better than others. I think most of you know that a cotton plant has a certain water potential to it as to how much water is in that plant and in that xylem to hydrate that plant, right? And that's based on the amount of roots it has and the amount of water that's in that soil volume that's able to explore. So we did a study. I had a couple graduate students. We went out. They killed me because we had to go out and do what's called pre-dawn water potential. So we went out at 3 a.m. and walked around with a pressure bomb and taking leaves of cotton fields that were highly mycorrhizal and those that were not. And then we went out at solar noon and did the same thing. We looked out, so what we did is collected a bunch of leaves, put them in a pressure bomb, looked at the water potential of the plant in those 2 time periods. And here's what we discovered. At solar noon, there was no difference between a field that had good mycorrhizae and a field that had almost no mycorrhizae. None. But you went out and looked at pre-dawn water potentials, and the plants that were mycorrhizal were completely hydrated again, and the plants that had no mycorrhizae were still suffering a 50% deficit from the day before. And that's the issue. The mycorrhizae can't solve the temperature response problem here and the amount of solar input that comes in. What they do is they allow the plant to recover better and faster if you have them. So as a consequence, the plant's going to keep going in the morning when the other plants that are stressed are still shut down. And that's what these fungi, we'll see later, that's what these fungi are able to do. Good questions. Okay, I'm going to go quickly through this just so you know. I'm going to talk about groups of fungi and what they are, just so you know, because you have to understand in terms of some of these broad taxonomic terms. So we have the zygomycota, which, and again, these are fungi that are growing in your system. They are all fast-growing. They're all saprophytic, but these fungi, and they produce a ton of spores. This is what I mentioned before as bread mold. These are the things that grow on your strawberries if you leave them in your refrigerator too long. They like that. So they're a fast-growing organism. They grow on the organic matter that's in the system. Here's what they look like as they're growing. But the thing about them is they cannot decompose any complex carbon. These are what are called secondary sugar fungi. They only can grow on simple sugar. So are they out in your field? Yes. Can you measure them as fungal mycelium and different things? Yes. Do they contribute to biomass? Yes. Are they doing anything to decomposition? Absolutely not. And so the problem is field A has a lot of zygomycetes in them and field B doesn't. Which one's doing better? Not field A. It can't do anything with build organic matter. So again, you have to keep in mind that not all fungi have the capability. The other major group are the ascomycetes, the ascomycota. And again, I'm putting this up because people should be familiar. These are the ones that are truffles, that form truffles, right? Or morels. But they're also the pathogen. Fusarium is an ascomycete. That's what's contributing to fusarium root rot, right? But they are the largest group of fungi, and they do contribute significantly to what's going on decomposition-wise. And again, take a look in your field. I've seen these in a number of stubble-managed fields around the Lubbock. These are cup fungi. These are what are called apothecia of how these fungi produce spores. They produce these cups with these sausage-like structures with spores in them. That's how they move around your fields. But I've seen them now. How big are they? A pencil eraser. They're not very big, right? But that's just the fruiting structure. That's not all the mycelium of these organisms. So that's one type. These are another. These are little flask-shaped structures that grow on your litter. They produce spores, and some of these are pathogens, right? And they'll look like that also. If you really want to have a really neat experience sometime, go out and simply collect some stubble out of your field. Put it, if you have a little microscope or magnifying glass, just put it into a Tupperware container. Keep it moist, not wet. Keep it moist, and just look at it over the next 3 months. You'll be amazed all the fungal structures that grow on it. And again, this is what's growing in your fields too. And then this is another one. These are, again, they're all ascomycetes, right? They all produce these little sausage structures with spores. These tend to be on plant leaves and whatnot. But this is how these fungi actually grow and reproduce in your fields. Now, the other thing that they do is a lot of these have an asexual stage. These are what we call the molds. This is Aspergillus. This is Phylophora. You know, this is what you're eating when you're eating blue cheese. You're eating all of these spores that people have put into the cheese to make it that color. That's what you're eating. This is what we're breathing in. By the way, this is what causes black mold. This is what's rotting out your plasterboard if it gets wet. But these fungi, these ascomycetes, also produce these structures that are growing around, and you're never going to see them naturally. But these are what are called deuteromycetes, but they're all the anamorphic stage of ascomycetes. And so here's some examples that you should know of. Penicillin, Aspergillus, Cladosporium alternaria. These are the ones that are important in terms of our environment. Now, what about cotton? Well, cotton leaf spot. So Bipolaris, Cercospora, Chronospora, you know Fusarium, you also know Verticillium. Those are ascomycetes. Those are no different. It's just that they're a pathogenic ascomycete. But you probably have, you know, this is why when you do sequencing of a soil, you say, well, the largest group are ascomycetes. Well, yeah, because they have the most species. That's what you get. It's going to be crucial to find out what genera you have to understand what your system's really doing. But they're the largest group. By the way, this causes us athlete's foot. Athlete's foot's an ascomycete. Ringworm's an ascomycete. Women who get vaginal infections, you're colonized by an ascomycetes yeast. That's why ascomycetes are one of the dominant groups around us. But they're carrying out the important part of decomposition in your fields. And then we get to the mushrooms, the basidiomycetes. And again, the basidiomycete, the mushroom, is the teleomorph. It's the one that is producing spores. But it's also, as with ascomycetes, it's a major decomposer of cellulose and lignin. These are the fungi that are really decomposing any organic matter that's contributing to soil organic matter formation. The problem with manure from my perspective, is it has too much soluble carbon. And I apologize if I'm saying something against someone else. I would never put molasses on a field. What are you feeding? You're feeding your secondary sugar fungi. You're feeding all your zygomycetes, and all they're going to do is gobble up that molasses and don't contribute anything to that field. Manure has too much dissolved carbon in it. And what's going to use it? Not the ascomycetes or basidiomycetes, it's the zygos.
Say that again about the molasses. Slow her down.
Sure. So the problem with molasses is it going to increase respiration activity? And the answer is yes, it'll prime it. But because zygomycetes are in that field, and it's a readily utilizable carbon source, the fungi that are not going to contribute anything to organic matter formation are going to use it first. So all those what are called, all the secondary sugar fungi, which are the zygomycota, are all going to get in there before the ascomycetes and the basidiomycetes can do anything. I mean, can any fungus use molasses? And the answer is yes. But which ones are going to get there first? The ones that are designed to get there first, and they're not the most efficient. Those are the weedy fungi.
So this is important to a lot of people in the room, trying to make notes of that. Even though they don't contribute to organic carbon, do they serve any other good functions, or are they these weedy fungi that are going to ultimately hurt us?
No, it's not that they're going to hurt. They're not going to benefit you in ways in which it's going to build organic matter in your soil. Now, the molasses, the manure, also allows them to grow. And so it's the nematodes, the protozoans, the mites that are going to eat them, too, if they're in large numbers. But you got to have the nematodes, the mites, and the other stuff that you have to have a good, mature soil food web in there, which means no tillage, no pesticides, no herbicides, because that's going to kill all your small soil animals also. So it's not one or the other. It's the complexity of how that food web, what eats what's able to grow. Anytime you put carbon, again, guys, I had a fellow student, friend of mine, that there's a whole group of fungi that are called urea fungi. They will only come up where there's urea. And they will grow. And the reason is they're designed to be able to deal with the conditions urea produces. And so you know what he used to do? He used to go and just collect his own urine, drink a lot of beer and get the urine and spread it around his fields, right? And look at these fungi. Again, this story is crazy, but you can always go out if you're hiking in the Rockies and you're at the right time, you can always go out and tell where a moose pissed, because you'll find a whole group of fungi, those apothecial ascomycetes, fruiting on it, because those are the urea fungi, right? And it's just the nature of how fungi use carbon and nitrogen. The problem, RN, with this whole notion of what happens is it's more complex than just what the fungi are doing. You have to take into account how the carbon nutrients, so again, those secondary sugar fungi are using the carbon you put on. Where are they getting the nitrogen from? It's from your soil. It's from your fertilizer. And what are they building for you? No, they're the squatters. Absolutely nothing. They're not contributing anything. Sir?
Organic world, at times, molasses is applied kind of as a PGR to slow the plant down. I don't understand, what's...
Well, it may slow it down because it's taking nitrogen from the plant and feeding the organisms. It's not just the fungi that are going to, this is a fungal talk. It's all the bacteria responding it too. Bacteria like to gobble carbon. And if you have enough nitrogen, they'll gobble all the carbon you want. But they're just not going to contribute a lot. Right. Yep. Yeah. There's always been this discussion in what's called the restoration literature about priming the system with glucose or priming it with molasses. And it'll work for a little bit. But it won't. But then next year you're back at the same place you were.
Okay, so that's why if you put it in your seed trench, it's going to work, because it's going to work for a little flashbang. But you're talking about you should put carbon over the whole thing. That's where you're messing up the system.
Yes. Yeah. Now the question is, why would you want to put it in there? So what are you priming in your seed trench? For what? And I'm serious about that. That's the notion. But what bacteria are you trying to prime? If your system had enough carbon in it to begin with, you wouldn't have to prime them.
Fish is a much better solution.
Yeah. Yeah. Fish.
Because
You-
that's a fungal food versus...
Yeah. And well, part of it is this. You want to have something that's a little bit more harder to decompose, because that's what's going to build the stuff. Now, if you're looking for just a jump start, what do they call the cans of stuff with 30% caffeine in them? For that we drink? Energy drinks. Yeah. You can give these things an energy drink. And this, again, the question is, if you need to do it, something's wrong with your system.
Something's wrong with our system.
Yeah. That's right. Something's wrong with the system. I'm drinking my coffee because I woke up at 3 o'clock this morning and couldn't fall back asleep. Right? Okay, let me finish this up. I think I'm almost done. Again, if you've ever looked at fungi, they have these gills that produce the spores. Right? And this is what you look at underneath a mushroom. That's what it looks like. Here's the gills, and here's all the basidiospores that get blown around. Now, I mention this because I've talked about this before. If you have mushrooms in your field, that's a good thing. Because what you know, and it goes back to that thing about fungal farming. If you have mushrooms in your field, you know that the organisms that are necessary to build soil carbon are there. And what you're doing is not detrimental. So current approaches, deleterious effects in soil fungi occur resulting cumulative. And again, this is out of some publication. It's not just any one year. It's a cumulative effect of what you're doing over time is having the impact. And the other part is too. It gets back to this silver bullet. You can't quickly add these back in any given growing season. It's going to take time to reestablish the interconnections between the plants, the carbon, and the fungi. And that's it for this first session. So I think I'm at, I'm a little over, but thank you guys. Good conversation. So we have time to open up for questions. Any further questions? Yes, sir.
Old days, planting fuzzy seed. Delanning is killing all of the life on the seed. The volunteer cotton and its vigor, just a comparison of the preload of what it has sitting there, a little thought.
Yeah. That's a good question as to how. I suspect this is part of the area we probably don't know. We don't ever know, as seeds germinate, how the plants pick up a lot of their microbiome. It does take a little bit of time, right? It's like an infant being birthed for us. We have to snuggle it, the mother has to feed it. That's how we do it. So rarely are the fungi on the seed coat and the bacteria the same ones that are on the roots. They're the ones from the top part of the plant. The top part of the plant and its microbiome is very different than the microbiome below ground because it's just different conditions. So I don't think that's necessarily the case. It's, more than likely it's the effect of all the chemical treatment of that seed has had an effect in terms of what it does. Now, having said that, if you have lint on a seed and all the volunteer cotton, the fungi that are decomposing that lint are creating nutrients that are around that seed as it germinates, immediately around that seed. And that's how all seeds function in nature to begin with. And that's probably what more is going on. It's all a different scale of what happens around that seed coat. Jim.
So some of my research sites have been continuous cotton, continuous tillage for 50 years or more. And I've seen some studies with the Kellogg Soil Institute that it can take, like they have a study looking at regeneration, looking at the microbiome, and they're at year 20, and they're only about 50% of the way to what would look like that native before
Right.
it's tilled.
Right.
Do we ever hit a point, especially here with our heat and our lack of moisture, that maybe we can't recover even if we go into these? Because I'm comparing this to some no-tills in the same area, and they're very slow to respond. They've been 10, 15 years, and we're not seeing the same increase.
increase.
We've damaged it too much that we can't come back.
No.
Maybe in my generation.
And that's all part of this question. And RN's brought this up too. Can we start building soil organic matter to the time we're not going to have enough irrigation capacity? So have soil organic matter be the way to store water, right? And I think the answer is it's not going to be in a couple years, right? It may be a decade, right? But it's going to be enough. But again, if you change it 1.1, 0.2, 0.3%, that's going to be sufficient enough to start making that difference over time. Now, the challenge for us is we don't have a good understanding of how warm winters and hot summers are going to affect that capacity going forward. We know what the climate models are indicating for us, right? That's right. That really is the dilemma as to whether or not, if we had same conditions back in the 70s going forward, no problem. We really don't know. But
We get the heat to increase our enzyme activity, but then we have no moisture.
right. No, right, yeah. And again, my take is, now I'm always optimistic. My take is if you can even when it's 120 degrees out or 110 degrees out, if you've shaded that soil sufficiently, you will have capacity with when you get any moisture to do what's necessary. And that's how you keep building it. Yes, sir.
So I had a question as far as, the past 2 days we've been talking about no-till and increasing organic matter and all that stuff. And part of the way we do that is the cover crops we put in and the root structures are there, and then we terminate those covers, and then we plant our cash crop on top of it. And those root structures that are now in the soil are contributing to organic matter.
Right.
So my thought with this fungus is, they have a branching root structure type thing going on. When we break or kill those, whether it's by having spray herbicide, pesticide, or something on top of that, and we kill that fungus, are those branching structures from the fungi, is that also contributing to the organic matter like the roots would be from the cover crop?
Yeah, well, yes, it is. So I'll go through this afternoon. In order to build long-term soil organic matter, you have to have certain breakdown products from the plant, but you also have to have compounds produced by the fungi. You have to have both, and that's why it matters what fungi are there. And if you don't have both, then you'll never burn. So what you'll do is you'll build organic matter that any organisms can use quickly within that season. You won't build long-term. And so the reason it's taking 50 years. Now, again, the Great Plains had far more organic matter then. So it didn't take, it took 50,000 years for them to get to there. But doesn't mean that we can't move the needle past 2% here, maybe to 2.5 within 8 years, a decade, depending upon. Yeah, you can, that's feasible. we're probably never going to get to 5 to 6 to 8. We just can't do it because of climate constraints, right? But as long as those plant roots are there, the fungi are going to use them, and they're going to. Now, the mycorrhizal fungi that were on the wheat that you terminated, they don't last long without a plant host there because they're not saprophytic. So unless you put something back that's going to allow them to recolonize something else, they won't last past 4 or 5 months. They're going to be gone. That were in those roots. Those roots are going to decompose and they're going to die. And everything will be degraded, but the mycorrhizal inoculum levels won't be where they were before you terminated that wheat. Now, the other question is, and something most of you can answer, which I don't know, when you terminate wheat, how much gets translocated below ground? I don't know. I don't think anybody's looked at that, right? How much of that herbicide gets translocated below ground? That's a problem. Yes, sir.
I assist farmers. I'm an ag consultant. And every year we pull soil samples. And I've had soil samples back 35, 40 years.
Can you speak up?
I work with farmers pulling soil samples every year and, try to tailor the recommendations based on what the soil lab reports and parts per million nitrogen
Right.
and that type of thing. And I always have the question, typically, if you read what Dan Krieg says or what the Extension Service says or soil scientists, say they recommend generally 40 to 50 units of nitrogen for every bale that you want to produce. And then there's also a similar level for phosphorus. And so, with my clients, we try to work out, what are their reasonable yield expectations and what type of water they have and able to produce those yields. But seldom do we worry about or consider, is a field a no-till field or is it a conventional till field? How long has it been in no-till? And I'm wondering if, our kind of hard and fast guidelines of 40 to 50 units, are those still accurate? Should they be, should you reconsider, nutrient level requirements?
So, I'm going to talk from strictly a fungal ecology, soil ecology perspective. They're probably outdated because those guidelines, as far as I know, are based on tillage systems, right? And the question is, and again, the question is, the nitrogen in your system has to do two things. It has to provide sufficient nitrogen to grow your crop. It also has to provide sufficient nitrogen for your microbes to live. And so you're balancing two out. Now, if you over-fertilize, the plant's only going to use a certain amount. It's not going to take up more, right, per unit time. But your bacteria will. And your fungi that are weedy fungi will. They'll keep using it and using it and using it. And so part of the dilemma is figuring out how much is really going to the plant and how much in excess are you just putting on, because that's the requirement. But the issue is, it's not ever going to the plant. It's going to the microbes that are going to gobble up carbon because you've given them the nitrogen to be able to do it. Because in natural systems, we'll see something this later this afternoon. In natural systems don't spike in nitrogen. They are continuous. And that's because of the recycling of that carbon, organic carbon, and the mineralization of that is uniform over a period of time. Now, there's going to be a spike when certain things happen, right, in the fall or whatnot, when you have all this flush. But mostly, systems don't change. I have to say, the limited data I have on looking at nitrate and ammonium levels and microbial responses is really from RN's fields, in terms of no-till. When I look at RN's fields compared to a conventional system, RN's nitrogen levels are low and they're pretty stable across the year, irrespective of what that crop is doing. That, for me, ecologically, is mimicking a natural system. The conventional system is spiking all over the place. And again, we just finished up with some hemp producers up past Dumas, looking at some mycorrhizal relationships. And so we did a bunch of soil, did some soil analysis. The producers didn't do any analysis. We had to do it for them. So we did it. We collected the soil in September, just prior to harvest of that hemp. One field, which was conventional, 5 of the samples had 130 parts per million of nitrate nitrogen in them. And I'm saying to myself, why would they, whatever they fertilized with, nothing ever used it. It just sat there to be leached out. So, I think that is, Bob, I think you're spot on. That's a question that has to be asked. How much nitrogen do you really need? I mean, if you were doing tillage and, again, you're putting on nitrogen at that rate because you have to do that every year. The question is, you don't want to do that every year. You want a system that's able to take the nitrogen that may have started out and then keep recycling it back. Now, again, because we cart stuff off, right? There's a certain amount of nitrogen that has to grow the bolls, and you lose. So you're going to have to put some back, but then again, that's where legumes come in. It's a question of how much to balance that out.
In Europe, they're actually, prohibiting farmers from using a lot of nitrogen.
Sure.
And, with our cotton production, you can't make up for nitrogen that was inadequate prior to planting after it gets into squaring because
it's too late.
Yeah, too late.
It's too late.
And so, I listen to different people talk about nitrogen requirements, and I sometimes wonder, some of the critics of fertilization, I think assume that growers are all over fertilizing. Maybe those that don't want nitrogen in the streams or the groundwater or whatever, I think they assume that all farmers are over fertilizing. But I look at my growers and I'd say, we're trying to cut it pretty close. And so I'm always asking myself, am I just right on? Because if anything, I'm probably, looking at those parts per million nitrogen, I could be shorting them a little bit.
Right. So let me ask a question to all of you. And again, I know we as researchers do this. I don't know how many of you do this. So all of you get soil tests, right? You all go out, you send stuff up. Somebody tells you how much ammonium, nitrate, phosphorus, calcium, magnesium. Those are spot measurements. That tells you at the time you sampled what was available in that soil pool. How many of you measure mineralization rates? No?
So we take the traditional soil test, and we've done some of the other tests over time, and I'm not smart enough to understand it, but, I'm sitting next to Clay, I'm gonna get myself in trouble here. He sends the same recommendations that you send.
Sure.
And he makes them based on the same numbers that you make. And in our cotton, for the last 4 growing seasons, we have used nothing but ammonium sulfate, and we kind of have a whole different fertility system. There's some both minute and nuts. But based on his recommendations, we're applying those 25% of what he recommends.
Mm-hmm.
So he's recommending this many units, we're putting out 25% of that in the form of ammonium sulfate, and that's it.
Yep.
And I don't think our yields are limited by nitrogen, but I can promise you that myself included, there's not a farmer in here except Carl Butcher. If you're fertilizing, it's a safe assumption that you're over.
Fertilizing.
Even me. But we don't know how these systems work. Well, Dr. Zach, John, I'm working on that.
Yeah.
John understands it more than most, but all of that work was done in the 1950s.
Right.
In dead soils that had been stored in a bunker for 6 months, homogenized, were completely void and absent of biology. It was a chemistry set. And that's just not the way ecosystems function.
Right.
And I am so happy to be here today. It wasn't great joy for me the last 2 days. There was a lot of great joy, but there was a lot of stress and pressure too.
Sure.
But this is great story. And I'm gonna try to soak up as much as I can, and then I'm gonna listen to the rest.
Okay.
and replay.
So one of the things we did with RN, and those of you that have gotten master's degrees and appreciate this, it was a lot of work, and the graduate students hated it. But they actually sell, and you can buy from a series of companies these little membrane filters that you take, and what you do is you activate them, you stick them in the ground, and what they are is they're ion exchange resin filters. And what they do is they measure the amount of, you have to have two different ones. They measure the amount of ammonium, and they measure the amount of nitrate that is moving through your system based on the mineralization of the organic matter by the fungi and the bacteria. And that's the amount of nitrogen that is generated per unit time by your system. Not what you applied, but by your system. And we've done this at Big Bend. And so one of the things that we've discovered, we have the little limited data we have with RN system is always generating nitrogen.
Tell me what you were calling this membrane. It's like a resin ball.
No, what they are, they're called membrane filters. And we had a company out of Canada we used to work with. I don't even know if they're still in business, but they look like throat depressors, and what you go to a doctor, he says, well, I'm going to check your throat, and he sticks a tongue depressor in. They're about that size, and they're coated, and so what you do is you pay them to ship them to you, you stick them in, you send them back, and you collect them. We always left them out for a month. And what it did is, every time it rained, every time it had soil moisture, you knew how much nitrogen per unit time was moving through your system as ammonium and nitrate. And I knew when RN would fertilize. And I knew when different things were going on because I could see it in the mineralization rate that comes through.
So I have a question for like RN and Bob. I know in some of the other states when they have these soil health plans and like nutrient analysis, they give credits, like nutrient credits for if you're no-tilling or if you're using a cover crop versus if it's a legume or a grass. Do we have that in the state of Texas?
I've never heard of anything like that, but, you know, a lot of work that Dr. Jack was doing, and when he was saying, telling us fertilizer, there was a time, we were growing off corn.
Right.
And we were putting 250 units of UAN through the pit a year. And the last corn crop we grew, we had 400 pounds of ammonium sulfate down. And this was the late planted stuff. And it made two numbers. It was planted 21st June, which just was a tough year. But I only put 20 units in season of UAN. So I still put a minute amount of UAN, but that's 100 units total on 200 bushels.
I can just speak like knowing NDSU and like South Dakota State Extension and that they'll go through and they'll ask, are you planting a legume as cover crop? Okay, well, that'll give you 50 units of nitrogen credit. So if you said you need this and you have this cover crop, then they're giving you this credit. And then if you're no-tilling, they also give you this credit. And it goes by what region of the state you're in, just because their climate changes from the west to east. So I just was curious if we did that here and got any kind of credit to take into account some of these other cover crops and management practices that we're using. So I hadn't seen anything, and so that's why I was wondering. I know you guys deal with that every year.
I think that I was really hurting the soil and the system, and I didn't mention that, but when I was looking through, and feel free, you beat me up too. I mean, because I've done so many things wrong, and you're so generous and kind, but you're not going to hurt my feelings if you want to talk about my moron complex.
No.
And some of the things that you might have witnessed.
Yeah. So no, and one of the things too, we need to take a break here, it's scheduled, but one of the things I would say, I'll ask you a question too. When you do a soil test, how often do you do it?
Once a year.
When I work on RN's fields, once a month.
Right.
I collect data for 6 years, once a month. And that's the only way to know what the dynamics are in that field. Once a year, forget about that.
I'll tell you, most times in season when I'm doing a tissue sample, I'm doing a soil sample at the same time.
Sure, yeah, so if you're...
Because I'm trying to
Yeah, you need to know what's going on over that, because as you go from April to, or February to March to April to May, souls are warming up, microbes are doing things, you're doing things, the plants are doing things. The dynamics of that system are completely changing over that period of time. And so what I would tell you is this: the limited data we have in RN's field for mineralization rates is that he has enough nitrogen moving through the system naturally by the microbes decomposing than adding any fertilizer. Now, are they going to do it every month? And the answer is no. That's part of the difficulty, because it's going to depend upon temperature and moisture, right, and what's going on. But he has enough nitrogen moving through naturally. And so one of the things, we do in research purposes. I mean, if I get on and talk to Katie Lewis's field, we're doing the same thing down in La Mesa that we're doing with anything else, or I do with Big Bend, right? We want to know how much nitrogen is actually moving through the system, not what's there as a static measurement. And again, because you're using the static measurement to tell you how much you have to add, it doesn't tell you how much actually moves through the system collectively over that year. And that's the difficulty.